Now, previously we have studied genetics, Mendelian genetics, and understood cell division. I think the information that you have gathered from both those two chapters will help you understand the modern genetics. Modern genetics is very recent. It was actually in the late 50s and early 60s that a lot of these discoveries were made, and the modern genetics has actually changed biology since then. Biology used to be the study of plants and animals only, and once the structure of DNA was determined, biologists gave the importance to a lot of the composition within the living cells. Um, so here I sort of introduced you to the term DNA earlier in the semester when we were studying chemistry, but here we will go into a lot more depth about DNA, its structure, and what is this composed of, and how it must undergo replication and undergoes mutations, and so forth. So let's go on with the discovery of this gigantic molecule. What does DNA do? That was the first question we need to explore. We have the knowledge of the chemistry of DNA, and we know that it's a genetic material. But how was the study taken place? Now, the study has been explained by Griffith in his very famous transformation experiment. Griffith used, he was actually, he was actually a European scientist, and I think he was mainly German, and he was a bacteriologist who studied bacteria. And he was trying to develop a vaccine against Streptococcus pneumonia, which causes pneumonia in mammals. And in 1931, which is really early in the 19th century, he performed a classic experiment with the bacterium. And he noticed that when these bacteria are grown on culture plates, he called a strain as the S strain. S strain is the strain that is the shiny, smooth colonies, and that's why he was using the term S, because the shape of the colonies were shiny, and they had a capsule, and which actually caused them to cause the disease. So he called that as the S strain. Now, there were bacteria that mutated and they did not have this capsule, and he called that as the R strain. Under the microscope, the S strain bacteria have a capsule, but the R strains do not. And when Griffith injected mice with the S strain of the bacteria, the mice died. And when he injected mice with the R strain, they do not die. So he said that the S strain is the virulent strain because it causes the disease, and the S strain is the non-virulent strain because it does not cause the disease and the bacteria, and the animal was able to live. Does that make sense so far? Because on these two experiments, he's shown to inject mice with the R strain, and then the mice with S strain is up here, and the R strain is in this example, and he shows you the virulent and the non-virulent strain. He then injected the mice with the heat-killed S strain. Now remember, the S strain is the one that causes the virulent strain. But when he did the heat-killed, he found that the mice does not die. How do you explain this? Heat-killed S strain does not cause the mice to die. Finally, he then took another combination in which he had injected the mice with a mixture of heat-killed S strain and the live R strain. Now remember, the live R strain also was non-virulent. So he took a mixture of both and he injected that, and he discovered that the mice dies. So he thus concluded that somehow some substance was necessary to synthesize the capsule, and therefore the virulence nature appeared, and somehow the virulent nature was passed from the dead S strain to the living R strain. So something from here actually passed on to to the non-virulent strain, and this caused the change in the phenotype of the bacteria, and which genetically then caused a change. And he at this time just used the term it's a genetic material. He did not know that this genetic material was DNA. He first introduced the term genetic material, that there was some substance that caused this change. This is the transforming change, because it transformed the S strain into the virulent strain. Now, these experiments later on were proved by other scientists, and we call them as the classic example of what DNA does. That is, DNA is the transforming substance. So, there were some other experiments that were carried out to confirm that whether DNA was the transforming substance. And one of the examples is an example by Hershey and Chase. Hershey and Chase were studying bacteriophages. Bacteriophages are viruses that infect bacteria only, and some of these viruses actually infect, for example, E. coli. So here's a structure of a simple bacteriophage. It has a head, which you can see is a big six-sided shape up here. And within the head is the DNA, or the substance, which is a genetic substance, along with certain and then outside is a coat, which is called as a protein coat, and it has prongs like tail fibers that help it to anchor on the surface of the E. coli cell wall, or the cell membrane. So very, very simple structure, only has a protein coat, which is the capsid, and this protein coat is surrounded by the DNA, which just enters the cell through this opening in the tail fiber, and it latches into the bacterial inside of the bacteria and divides within that, and then eventually will cause more new phages to be produced within the bacteria. So this helped them, because we have the protein, which is in the coat, and we have the genetic material, which is the DNA. So there are only two things. And if the experiment shows that the coat and the tail fibers are all left behind when the bacteria infects when the virus infects the bacteria, and the only thing that goes in is the DNA, and then the DNA causes this change to produce more bacteriophages. Now, how they did that was they used radioactive elements in there. They used radioactive DNA, which is the P32, and they used, in a second experiment, sulfur 32. Now, sulfur is a radioactive element, and so is P32. Now, later on, once they had, in both experiments, I'm going to talk to you firstly about the first experiments. Now, when the bacteria and viruses are cultured together, the radioactive DNA enters the bacteria, and within it starts dividing within the bacteria and produces no more bacteriophages. Now, after mechanical agitation and dislodging the cell and actually putting it in a centrifuge, something that some of you may have done that in one of my experiments in class. A centrifuge is a big apparatus or a gadget that actually spins the tubes, and they settle down based on sediment, on how heavy they are. So bacteria's if the sediments are, if the radioactive is present in the sediment, it was considered to be because of DNA, because there are more DNA up here, and that's why it was found that DNA must be the genetic substance. In a parallel experiment, they did the same thing with the sulfur. Now, the sulfur coat leaves behind. So when they do the same thing, we let it grow, we take it out, we centrifuge it, agitate it, and we do the sediment. Big viruses in this are liquid are only on the top are radioactive, bacteria and the sediment are not radioactive. So this tells you that if the radioactivity did not enter the bacteria, then the protein did not enter the bacteria. What entered the bacteria was the DNA or the starting of the genetic material which entered the bacteria. So this experiment pretty much confirmed the proposal by Griffith from his transformation experiment. Now that we have understood that, okay, DNA is the starting material, what is the genetic substance or transforming material, what is it made up of? Now recall, I did this earlier in the semester. The DNA is made up of something called as a nucleotide. A nucleotide has 3 parts. Recall that we have a central region which is called as the ribose sugar. We have the phosphate group, and on the other side we have the nitrogenous bases. So you have 3 parts. The ribose sugar is always going to be present in all the DNA molecules. The phosphate will always be present. However, the nitrogenous bases are of 4 different kinds. The 4 different kinds of nitrogenous bases can actually be divided into 2 types. One is the purine, the second one is the pyrimidine. The purine is the double ring. So the bases that appear to be double ring, for example, this one, we have 1 ring here, we have a second ring here, we have 1 ring here, we have a second ring here. These 2 are your purine bases. On the other hand, pyrimidine bases are the single ring, and these are thymine and cytosine. So we have either you can have a DNA nucleotide which is adenine nucleotide, or you can have a guanine nucleotide, or you can have a thymine nucleotide, or you can have a cytosine nucleotide. So this is some of the terms that I did earlier in the semester. I just wanted to go back and review that with you. So you have purine nucleotide or a pyrimidine nucleotide. Now within the purine and the pyrimidine nucleotide, scientists later on used the term complementary, which meant that one of these bases will only pair up with pyrimidine bases. Now the reason, a little background I want to give you on this one is when the structure of DNA was being studied, DNA structure was shown to be a uniform width. So if it's a uniform width, and some of the experiments that we will talk about later will show you that the sugar phosphate forms these the stem of this ladder, and the steps of the ladder are actually made up of these nitrogenous bases. So in order for the steps of the bases to be uniform width, a purine had to pair up with a pyrimidine. Does that make sense to you? A double ring with a single ring? Because if I have 2 double rings, then perhaps the width would be wide, and then it would be narrow, and then it would be wide, and so forth. And that did not make sense. So I'm going to clean a little bit of this slide. It's getting a little bit messy. Sometimes I don't like it. Okay, so for the width of the DNA to be uniform, you had to have one double ring paired up with a single ring to keep the width uniform. And this is called as complementary base pairing, which means that a purine always bounds to a pyrimidine. Now, which purine bounds to which pyrimidine was the next question, which I will answer you in a couple of slides later to see how which purine was forced to pair up with which pyrimidine. So, but we will get into this a little bit later. Actually, you know what? I'm just going to write it over here, and we will go ahead and redo it again. Adenine and thymine were always shown to have the same amount. So if you notice over here, adenine, thymine, guanine and cytosine. So guanine and cytosine were the same amount, and so was adenine and thymine. So notice up here, if we have 25% of guanine, you have the same amount of cytosine, and then the opposite up here. So pretty much if you know the amount of adenine, let's say in an example I say adenine is 15%, and I want to know how much cytosine is present. Can you do that? Now think about it. If adenine is 15%, then thiamine is also going to be 15%. That takes care of total of 30%. What remains is 70%. So half of that would be 35 cytosine, 35 guanine. So pretty much this is the concept of complementary base pairing. Now, it's really important to give tribute to Rosalind Franklin when we're studying the structure of DNA, because a lot of these technologies, or the first X-ray images that are shown in this picture over here, were taken by Rosalind Franklin. Rosalind Franklin had actually developed the technique of X-ray diffraction, in which a beam is diffracted to the crystals of DNA, and then the repeating patterns were seen in this image of here, which looks like an X-ray. Now, Rosalind Franklin actually passed away by the time Watson and Crick proposed the model for DNA, and in order to receive a Nobel Prize, you had to be living. So Watson and Crick are famous for their structure of DNA, but that was based on Rosalind Franklin's study. So Rosalind Franklin had given the X-ray images, which helped Watson and Crick, and also they actually also used information from Shargov. So there were like many different scientists that were working and putting in some data, which was all put together by Watson and Crick. They knew that DNA is a polymer of nucleotide, but they did not know how the nucleotides were arranged within the molecule. They proposed the double helix structures, in which they said the sugar phosphate forms the outside backbone of this double helix, and the steps or the paired bases form the inside of the double helix. And the paired bases is based on Shargov's information about how adenine and thiamine can only form 2 hydrogen bonds, and guanine and cytosine can have 3 hydrogen bonds, which is the basis of complementary base pairing. So now we know that the DNA double helix is a twisted ladder with the sugar phosphate making up the sides of the ladder, and the hydrogen bond, which makes the basis of these rungs or the steps of the ladder. So let's go on with our next image up here. In this next image, we are going to look at the same DNA model, and here are the hydrogen bonds showing between guanine and cytosine, and showing why the complementary base pairing was forced to be in this pattern. Now, another interesting thing I want you to point finger over here on this structure of DNA is the 5 versus 3 prime. Now, in the DNA molecule, one strand is has the 5 prime end over here, and notice you've got the 3 prime end over here. The strands are actually running anti-parallel. With complementary base pairing. So in this way, what it means is that one strand would have P on one end and 3 on the other. The other strand would have the anti-parallel, which would be 5 prime on this end and the 3 prime on this end. Now these two 5 prime and 3 prime ends are important, because we will talk later about how DNA replicates and how it's how the new strands are formed. So I want you to sort of remember what is the 5 and the 3 end. The 5 prime end is the end where this is the 5th carbon where the phosphate is attached, and the 3rd carbon is where it's attached on the bottom. So this is what the 5 and the 3 are representing, the 5 end and the 3 end. Notice that if it's an anti-parallel strand, you have the sugar flipped or in a mirror image up here. So you have the 5 on the bottom here and 5 on the top here. So it's basically based on the location of the carbon on the sugar. So 5 prime, 3 prime is is a terminal end that we use for representing the two strands. So I'm going to stop here and we will then later on understand the second section.